Slide 1. Peripheral circulation is a series of slide tape programs developed and produced by the American Physiological Society. This is Local Control Part 2. Content experts, Dr. Brian R. Duling and Dr. Paul C. Johnson. Please note that a printed study guide accompanies this program. It contains the full program text, a test of the concepts presented, and references for additional study. 2. In Part 1 of this program, we introduced the mechanisms contained wholly within tissues and organs that maintain the blood flow necessary to meet the needs of the tissues and enable them to carry out their functions. We discussed in detail the functional and mechanical characteristics of vascular smooth muscle and described its response to distension. 3. In this program, we describe the metabolic interactions between tissue and vasculature that balance blood flow and tissue function, and we analyze the combined effects of these metabolic mechanisms and the myogenic response in three major phenomena: functional hyperemia, reactive hyperemia, and autoregulation. 4. Your first objective is to be able to outline the basic interactions between tissue and the vasculature that balance blood flow and tissue function. 5. Metabolic regulation refers to any process that links the metabolic activity of either the vascular smooth muscle or the parenchymal cells of an organ to blood flow rate. The essence of the idea is that tissue metabolic activity contributes in some way to the environment of the vascular smooth muscle cells and thereby influences their contractile state. 6. For example, an increase in a tissue's metabolic rate may increase the concentration of a dilator metabolite such as carbon dioxide in the extracellular fluid. When the CO2 concentration increases, vascular smooth muscle cells relax, the arteriole dilates, and blood flow increases. The metabolic event, CO2 formation, is coupled to a vascular response, vasodilation. Note that one effect of the increased flow is to facilitate removal of the dilator metabolite. 7. Dilator metabolite concentration is determined by the ratio of dilator production to dilator removal. This relationship is an important factor in metabolic regulation. If the metabolic rate generates dilator substances that increase flow in proportion to the metabolic activity, the ratio between production and removal is constant. 8. The vasculature can facilitate removal of metabolic materials from the tissues by opening unperfused capillaries. This decreases the diffusion distance between the cells and the capillaries. 9. When intercapillary distance is reduced, the concentration of metabolites between capillaries does not rise as high, so the mean tissue level of metabolites is reduced. Changes in capillary density are usually seen during local regulatory responses. 10. The primary event in metabolic regulation may be a change in blood flow rather than a change in metabolism. If arterial pressure is reduced, for example, there is an initial reduction in blood flow. Then, since the dilator removal rate is reduced, vasodilator metabolites accumulate in the tissues, inducing vasodilation and restoring flow. 11. Graphically, the sequence is as follows. Pressure is reduced, flow declines, and tissue levels of metabolites increase. Metabolites cause smooth muscle relaxation, and flow is returned toward the original level. The increased flow increases dilator washout, and a new steady state is established. 12. Sketch a simple flow chart showing the metabolic control events in response to an increased metabolic rate. Please stop the tape while you answer this question and those that follow throughout the program. 13. Increased tissue metabolism changes the environment of the arteriolar smooth muscle cells, causing them to relax. Smooth muscle relaxation increases flow and the number of open capillaries, both of which increase capillary transport capacity. The elevated capillary transport capacity acts in conjunction with the elevated metabolism to stabilize tissue extracellular metabolite concentrations. 14. Your next objective is to be able to describe the sources of vasodilator metabolites that couple metabolism and blood flow. 15. Since most cellular functions ultimately obtain energy by splitting ATP, it follows logically that the vasodilator metabolites responsible for flow regulation might be tied closely to cellular energetics. 16. ATP hydrolysis in itself generates several vasodilators. These include phosphate, hydrogen ion, AMP, and adenosine. 17. The following regulatory scheme has been proposed. The tissue increases ATP hydrolysis in order to supply energy necessary for an accelerated cellular activity such as ion pumping or muscle contraction. The breakdown products of ATP are formed at an increased rate and diffuse into the vicinity of the vascular muscle. 18. As the dilators accumulate and the vessel relaxes, the rate of dilator removal increases. A new steady state is reached when the elevation in dilator washout equals the increment in dilator production induced by the initial increase in metabolic function. 19. Another source of vasodilator substances is linked to the fall in cellular levels of ATP during increased metabolic activity. The decrease in ATP concentration and the increase in the concentrations of its breakdown products, ADP and AMP, stimulate cellular metabolic processes that regenerate ATP. The processes responsible for ATP generation include glycolysis, shown here as the breakdown of glucose to pyruvate, the Krebs cycle, and the cytochrome chain. 20. When these processes are stimulated, a variety of intermediates accumulate. The most notable of these with regard to regulation of flow is CO2, which is a vasodilator metabolite of the Krebs cycle. In addition, hydrolysis of CO2 to form carbonic acid increases the concentration of hydrogen ion, which is also a vasodilator. 21. List the products of ATP hydrolysis that are likely to be involved in metabolic control of blood flow. What product of the Krebs cycle is likely to be involved in metabolic regulation of flow? 22. The products of ATP hydrolysis that are likely to be involved in metabolic control of blood flow are AMP, phosphate, hydrogen ion, and adenosine. The Krebs cycle enzymes produce CO2, which is a vasodilator. 23. Your next objective is to be able to describe two means by which oxygen may regulate blood flow in proportion to metabolic needs. 24. Oxygen has been linked to metabolic regulation through various mechanisms in a variety of tissues. As indicated by this graph, reduction of arterial oxygen content causes vasodilation. This is especially marked in the heart, brain, and skeletal muscles. Broadly speaking, the control of both arteriolar flow and capillary density can be related in part to tissue oxygen availability. 25. Oxygen availability may influence tissue blood flow by acting on either of two sites. It may act directly on the smooth muscle cells of the microvessels, or it may act indirectly by changing parenchymal cell metabolism. 26. In the hypothesis proposing a direct action of oxygen on the smooth muscle, it is assumed that the arteriolar smooth muscle relaxes when its oxygen tension is diminished. The regulatory scheme is simply that increased metabolism or decreased flow produces a decrease in smooth muscle PO2. The smooth muscle relaxes, causing vascular dilation and a compensatory increase in oxygen delivery. 27. The pivotal idea of the indirect action of oxygen on the smooth muscle is that the oxygen concentration in functioning cells determines the rate at which a vasoactive substance is produced. Most evidence indicates that a decreased PO2 allows formation of a vasodilator. The metabolite that couples tissue oxygen availability and vascular smooth muscle tone is not known with certainty, although there are a number of likely candidates. 28. The link is generally thought to be related to the process of oxidative phosphorylation and the elements of the cytochrome chain, which generate ATP. When tissue metabolism is stimulated, the demand for oxygen increases and the available oxygen is used to resynthesize ATP. If the oxygen supply is inadequate, the ATP synthetic rate is limited, and the metabolites of ATP accumulate, producing vasodilation and increasing oxygen delivery to the tissues. 29. In addition, when the oxygen concentration is low, the cells shift to anaerobic metabolism. This results in the formation of lactate and hydrogen ion. These vasodilators enhance the dilation already induced by the products related to ATP hydrolysis and the intermediates of ATP resynthesis. 30. Altered metabolism also leads to changes in tissue osmolality. Both ATP hydrolysis and the process of anaerobic metabolism lead to the formation of a larger number of particles than in the parent compounds. These particles increase tissue osmolality. Since hyperosmotic solutions produce vasodilation, the simple increase in particle number is an additional coupling of metabolism and blood flow. 31. At what two sites might oxygen act to control blood flow? 32. Oxygen might act directly on the vascular smooth muscle cells or indirectly on the parenchymal cells to induce the release of a vasoactive substance. 33. In the remainder of this program, we discuss three local control phenomena that arise from the combined actions of metabolic and myogenic mechanisms and which are found in almost all vascular beds. They are functional hyperemia, reactive hyperemia, and autoregulation. By examining these patterns of behavior, we can understand better how the peripheral circulation responds to tissue needs. 34. Your objective in this section is to be able to define and describe functional hyperemia. 35. Functional hyperemia is simply the adjustment of blood flow to the level of tissue activity. In a steady state, blood flow in skeletal muscle is regulated largely by the release of the vasodilator metabolites that mediate dilation in response to metabolic demands. During increased activity, blood flow is closely related to tissue oxygen consumption via the mechanisms we have just described. 36. At rest, skeletal muscle with a blood flow rate of 3 milliliters per minute per 100 grams tissue consumes about 30% of the oxygen supplied to it in the arterial blood. 37. When metabolism increases, the tissue first increases oxygen consumption from the available blood supply. 38. Functional hyperemia leads within seconds to increased blood flow. At a flow rate of 50 milliliters per minute per 100 grams tissue, the tissue may consume as much as 80% of the oxygen supplied. Here, blood flow and oxygen consumption are plotted during sustained skeletal muscle exercise. Oxygen consumption rises from 0.2 milliliters oxygen per minute at a flow rate of 3 milliliters per minute to 12 milliliters oxygen per minute at a flow rate of 60 milliliters per minute. Patterns similar to this are found in the myocardium. When the heart rate increases or the contractile force increases, the coronary blood flow and oxygen consumption rise. 40. The next few slides, we discuss several specialized mediators of dilation in functional hyperemia. Potassium may assume the role of a dilator metabolite in tissues where activity patterns are associated with changes in the electrical potential of the cells. During repolarization of cells following an action potential, there is an efflux of potassium. This potassium may accumulate in interstitial fluid and dilate the vessel, increasing oxygen delivery to the tissue and washing metabolites out. 41. As indicated in this plot of extracellular potassium concentration and blood flow, the role of potassium as a vasodilator is limited because its dilator effects are transient. Nevertheless, it is likely that potassium is an important initiator of functional hyperemia in striated muscle and brain because it provides an immediate vasodilator stimulus with the onset of increased activity. 42. Through the following sequence, a variety of organs and tissues undergo a very special type of functional hyperemia. First, neural elements are triggered that increase the tissue's metabolic rate and induce the release of an enzyme, kallikrein. Kallikrein acts on a protein substrate called kininogen, resulting in the formation of a decapeptide, kalladin. This can be split by enzymes present in the tissue, forming bradykinin, a nonapeptide. Kalladin and bradykinin are very potent vasodilators. This type of functional hyperemia is seen in exocrine glands, such as the pancreas, salivary glands, and sweat glands during periods of increased secretion, in the skin during functional dilation, and in the stomach and intestines during digestion. 43. Therefore, neural activation of the glands is automatically coupled with the formation of bradykinin and an appropriate vasodilation to match blood flow with the increased metabolic rate. 44. What are the two ways in which the tissues obtain more oxygen when metabolism increases? In what types of tissues might potassium play a role in functional dilation? 45. Tissues obtain more oxygen when metabolism increases by extracting more oxygen from the available blood and by increasing blood flow. Potassium may induce functional dilation in electrically active tissues as a result of its release during cellular repolarization. 46. Schematically outline the biochemical sequence involved in kalladin and bradykinin formation. 47. Neural elements trigger the release of the enzyme kallikrein, which acts on the protein substrate kininogen to form the decapeptide kalladin. Kalladin may be split enzymatically to form the nonapeptide bradykinin. Both kalladin and bradykinin are potent vasodilators. 48. Your next objective is to be able to define and describe reactive hyperemia. 49. Reactive hyperemia is an increase in blood flow to the tissues following a period of decreased flow or flow arrest. 50. Here, the response following partial occlusion of arterial inflow is represented graphically. A significant response may be seen after as little as 3 seconds of flow arrest. 51. The magnitude of the response varies greatly from organ to organ, but is seen to some extent in all tissues. Reactive hyperemia is especially pronounced in cardiac muscle, skeletal muscle, and skin, where flow may increase by a factor of 5. It is less prominent in the liver and gastrointestinal tract, where flow may double. It is weak in the kidney, where flow may increase by half. 52. The following evidence suggests that a metabolic mechanism accounts for reactive hyperemia. As the period of flow arrest increases, the peak and duration of the response both increase. The fact that the response is enhanced by longer occlusions suggests that vasodilator substances accumulate in the tissues and mediate the vasodilation. 53. It has also been suggested that loss of the normal myogenic contraction of the vascular smooth muscle cells causes reactive hyperemia. Recall that the myogenic response is vascular smooth muscle cell reaction to a stretching force. When intravascular pressure falls during partial arterial occlusion, the myogenic tone decreases. 54. When pressure is suddenly restored, active smooth muscle tone is low, allowing the arteriole to dilate and blood flow to increase. 55. Here is a comparison of responses following arterial occlusion and following venous occlusion. When venous outflow is stopped, reactive hyperemia is not as pronounced. 56. This is probably due to two factors. Since venous pressure is maintained during occlusion, the myogenic tone is maintained. And during venous occlusion, total organ blood volume is greater, which provides a greater reservoir of oxygen in the stagnant blood. 57. What are two proposed mechanisms of reactive hyperemia? Cite one piece of evidence in favor of each of the proposed mechanisms. 58. Two proposed mechanisms for reactive hyperemia are the accumulation of vasodilator metabolites and the loss of the pressure stimulus for the myogenic response. The increased duration of hyperemia with increased occlusion time favors a metabolic mechanism. The reduced hyperemia with venous occlusion favors a myogenic mechanism. 59. Your last objective is to be able to define and describe autoregulation of blood flow. 60. Autoregulation is the tendency for organ blood flow to remain constant by active vascular adjustments during variations in arterial pressure. Autoregulation is a ubiquitous phenomenon. It has been seen in virtually all tissues. 61. The nature of autoregulatory adjustments may be described graphically as a pressure step associated with the rapid increase in flow, which returns slowly to near normal levels. When blood pressure is suddenly increased, first the blood vessels expand passively and flow rises. Autoregulatory mechanisms then constrict the arterioles and bring flow back to control levels. The mechanisms responsible for autoregulation are thought to be the same as those responsible for reactive hyperemia. 62. First, there are changes in tissue metabolite concentration. The increased flow associated with the pressure step washes out vasodilator metabolites, allowing constriction. 63. Second, the pressure stimulus to the vascular smooth muscle triggers the myogenic response. 64. Conversely, the typical autoregulatory response to reduced arterial pressure is arteriolar relaxation and maintenance of constant blood flow. 65. Autoregulation occurs over a specific range of arterial pressure. In certain organs such as brain, kidney, and myocardium, the mechanism is so pronounced that blood flow is virtually unchanged over the pressure range from 80 to 160 millimeters mercury. 66. The lower limit of autoregulatory response is the point at which the arterioles are completely relaxed. This limit, shown here at 80 millimeters mercury, varies between 40 and 80 millimeters mercury in different organs. 67. As internal pressure drops below this limit, the relaxed vessels collapse passively. As a consequence, resistance rises and the flow falls drastically. 68. The upper limit occurs when the smooth muscle loses its ability to withstand the internal pressure. At this pressure, the contractile machinery is apparently unable to maintain its shortened state and length increases. This point is shown here at 160 millimeters mercury. It occurs normally within the arterial pressure range of 160 to 200 millimeters mercury. 69. As the autoregulatory mechanism maintains constant tissue blood flow, it also keeps capillary pressure constant. Recall that capillary pressure, PC, is equal to the product of blood flow, Q, times venous resistance, RV, plus venous pressure, PV. So long as flow is maintained, a fall in arterial pressure will not alter capillary pressure. 70. The importance of a constant capillary pressure is seen in an organ such as the kidney, where autoregulation helps maintain a constant filtration pressure in the glomerular capillaries. Within the autoregulatory range, the kidney's normal plasma filtering function is protected from a drop in arterial pressure. In organs such as liver and intestine, whose capillaries have a high filtration constant, autoregulation helps maintain a normal tissue fluid volume. Recall that given a high filtration constant, even a small change in capillary pressure leads rapidly to a change in tissue fluid volume. 71. The importance of autoregulation is also seen in the physiological response to coronary artery disease. In atherosclerosis of the major coronary arteries, the resistance to flow in the large arteries increases. 72. In response to reduced myocardial blood flow downstream, autoregulation maintains normal flow by dilating the arterioles. This maintains normal tissue perfusion until the atherosclerosis becomes so severe that the arterioles are maximally dilated. 73. When the pressure downstream from the obstruction has reached the low limit of the autoregulatory range, any further atherosclerosis leads to a disproportionate fall in flow because the arterioles collapse passively. 74. We should emphasize that local regulatory mechanisms may be opposed by neural and hormonal inputs. When there is a general systemic pressure reduction, for example, local regulatory mechanisms may be overridden by the sympathetic baroreceptor reflexes, such as the carotid reflex, which cause vasoconstriction and maintain arterial pressure. 75. Two organs are generally exempt from the carotid reflex. These are the brain and the myocardium. The fact that in response to a systemic pressure drop, other areas are constricted while the brain and myocardium are allowed to autoregulate assures that both receive blood flow in accordance with their metabolic needs. 76. Define autoregulation. What mechanisms are responsible for autoregulation? What determines the lower and upper limits of the autoregulatory range? 77. Autoregulation is the tendency for blood flow to remain constant despite changes in arterial pressure. Two mechanisms have been proposed: changes in tissue vasodilator metabolite concentrations and a myogenic response to increased intravascular pressure. The lower limit occurs when the vascular smooth muscle is completely relaxed. The upper limit occurs when internal pressure creates a wall tension greater than vascular smooth muscle can withstand. 78. This concludes the presentation of Local Control, Part 2. We have described the metabolic interactions between tissue and vasculature that balance blood flow and tissue function, and we have analyzed the combined effects of metabolic regulation and the myogenic response in three major phenomena: functional hyperemia, reactive hyperemia, and autoregulation. To assess your understanding of these concepts and for a list of additional readings, please refer to your study guide. 79. In the next program in this series, Neural Control, we discuss the sympathetic innervation of the vasculature, the synthesis and release of the sympathetic transmitter norepinephrine, the vascular smooth muscle response to sympathetic stimulation, the hemodynamic effects of nerve stimulation to an organ, and the effects of sympathetic activity on capillary exchange. 80. This program is part of the Peripheral Circulation Series. It was developed and produced by the American Physiological Society. Copyright 1981.